সেন্সেশন। এই প্র্যাকটিক্যালে এটা করেছো তুমি, তো বাংলা বুঝতে পারো তো, তাহলে এই জন্য বাংলাই বলি এখন, তো যখন আসেনি। টেস্ট সেন্সেশন, এটার রিসেপ্টর অর্গান কি টেস্ট সেন্সেশনের? প্র্যাকটিক্যাল কাজ তো করেছো। হ্যাঁ, রিসেপ্টর অর্গান কি? এটা টাং। টাঙের মধ্যে পার্টিকুলার রিসেপ্টরটা কি বলে? একে বলে টেস্ট বাডস। টেস্ট বাডস, টেস্ট বাডস, টেস্ট বাডস কোথায় কোথায় থাকে? কোথায় থাকে টেস্ট বাড? না, ওটা টেস্ট বাডটা কোন জায়গায় থাকে? টাঙে থাকে, কিন্তু টাঙে কোথায় থাকে? বলতে হবে টাঙের ডরসাল সারফেসে থাকে। ভেন্ট্রাল সারফেসে থেকে ইউটিলিটি নেই, সেই জন্য নেই। হ্যাঁ, ডরসাল সারফেসে। ডরসাল সারফেসে এমনি অন্য জায়গায় ছড়িয়ে থাকে না, বেশি অল্প পরিমাণ থাকে। এগুলো থাকে প্যাপিলাতে। প্যাপিলা, প্যাপিলার মধ্যে তোমার ফাঙ্গিফর্ম প্যাপিলা, ফাঙ্গিফর্ম, আর ভ্যালেট প্যাপিলাতে থাকে। ভ্যালেট, ফোলিয়েট প্যাপিলাতেও থাকে। ফোলিয়েট, কিন্তু ফিলিফর্ম প্যাপিলা, যেটা সংখ্যা বেশি, সেটাতে থাকে না। ফাঙ্গিফর্ম প্যাপিলাতে থাকে, ভ্যালেট প্যাপিলাতে থাকে, ফোলিয়েট প্যাপিলাতে থাকে। বুঝতে পারছো? এখানে প্যাপিলা, এই প্যাপিলাতে গুলোতে থাকে। ফাঙ্গিফর্ম নামটা হচ্ছে, নামটা থেকে বুঝতে পারছো। এই প্যাপিলা গুলো খালি চোখে দেখা যায়। খালি চোখে এই প্যাপিলা দেখা যায়। একটা হ্যান্ড লেন্স নিয়ে তুমি ইচ্ছা করলে এটা দেখতে পারবে। টাংটা ওপেন করে একটু যদি ফিল্টার পেপার দিয়ে শুকনো করে নাও, তারপরে দেখবে এগুলো পরিষ্কার বোঝা যায় প্যাপিলা। কিন্তু টেস্ট বাড খালি চোখে দেখা যায়। যেমন এর ক্ষেত্রে আপ টু 5, পাঁচটা অব্দি থাকে ফাঙ্গিফর্ম প্যাপিলাতে থাকে। ফাঙ্গিফর্ম প্যাপিলাতে কোথায় থাকে? এটা হচ্ছে অ্যান্টেরিয়র পার্ট অফ দি টাঙ্গে থাকে। ডিপ অ্যান্টেরিয়র পার্ট, কিছুটা ল্যাটারাল পার্টও থাকে। আর বেশি পরিমাণ থাকে হচ্ছে ভ্যালেট প্যাপিলা। ভ্যালেট প্যাপিলা, ভ্যালেট প্যাপিলা, ভ্যালেট প্যাপিলাতে মনে করো এরকম থাকে টাং, এরকম ভাবে একটা ভি শেপের থাকে। ভি শেপের এইভাবে থাকে প্যাপিলা, প্যাপিলাগুলো এখানে এইভাবে। এই প্যাপিলাগুলো এরকম ভাবে থাকে। 8টা 10টা 12টা প্যাপিলা থাকে। এটা মিড লাইন, ভ্যালেট প্যাপিলা। ভ্যালেট প্যাপিলাতে 100 বা তারও বেশি থাকতে পারে ভ্যালেট প্যাপিলা। ভ্যালেট প্যাপিলাগুলো স্ট্রাকচারটা হচ্ছে, এখান থেকে ভ্যালেট প্যাপিলার স্ট্রাকচারটা হচ্ছে তোমার এখানে এরকম থাকে, এবং তার পাশে একটা গ্রুপ থাকে এরকম ভাবে। এবং এর ওয়ালে, ইনার ওয়ালে, এসব জায়গায় প্যাপিলাগুলো থাকে। বুঝতে পারছো? এরকম। এটা হচ্ছে 100, 250 আপ টু, কোন বই বলছে মোর দেন 100 থাকছে, কোন বই বলছে অ্যানাটমি টেক্সট সেটা বলছে 250 অব্দি টেস্ট বার থাকতে পারে এখানে। আর ফোলিয়েট প্যাপিলাটা হচ্ছে এরকম জায়গায় থাকে, এইরকম। এই ফোলিয়েট প্যাপিলাটা ল্যাটারাল সাইডে পোস্টেরিয়র পার্টে থাকে, এবং এতেও থাকে, এবং এতেও তোমার টেস্ট বার থাকে ফোলিয়েট প্যাপিলা। এটা তো টেস্ট বার থাকে দু সাইড সব জায়গায় মিলিয়ে আপ টু 10,000 টেস্ট বার থাকতে পারে। হ্যাঁ, ফোলিয়েট প্যাপিলা, প্যাপিলা, প্যাপিলার মধ্যে কি থাকে, বার্ড থাকে। প্যাপিলাগুলি বললাম যে খালি চোখে দেখা যায়, তুমি বাংলা বুঝতে পারো না তো, বুঝতে পারো? হ্যাঁ, তাহলে পরে সেক্ষেত্রে প্যাপিলাগুলো তোমার খালি চোখে দেখা যায়, কিন্তু টেস্ট বার্ডগুলো খালি চোখে দেখা যায় না, তার জন্য মাইক্রোস্কোপের দরকার পড়ে। বুঝতে পেরেছ? বলে এখন কি আসবে না আসবে, এই জন্য আমাদের অভ্যাস মতো ইংরেজিতেই বলা উচিত। 10,000 taste buds. It is the organ of taste, taste buds. They are placed on the dorsal surface of the tongue, in the papilla, and to some extent outside the papilla also. And they are present in the fungiform papillae, valate papillae or circumvallate papillae, and also foliate papillae. In the fungiform papillae, they are numerous in the anterior part of the tongue near the tip, and they contain up to 5 taste buds. And valate papillae or circumvallate papillae, they are present in the V form, at the junction of the anterior 2/3 and posterior 1/3, and they are histologically, you will see this type of structure, and there is a valley, or there are some groove outside it, and then in the wall, in this wall, internal wall and external wall to some extent, they are more numerous in the internal wall, this taste buds. Besides this, there are some glands that secrete some substances. Ebner's gland, von Ebner's gland, and they secrete some fluid, and this fluid, similar to that of odorant binding protein secreted by the Bowman's gland in olfactory system, and they have some compound or proteins that can binding with taste producing substances, similar to the action of the odorant binding protein, that concentrate the testants or taste producing substances, and they contain taste buds even up to 100, and in anatomy text there is 250 per papilla, and foliate papilla, they contain their number, textbook does not mention the proper number, but they are intermediate between them, and they are in the, and foliate papilla, it is part of anatomy also, and they are in the lateral side, and in their walls, especially posteriorly, there are taste buds. Their exact numbers not given in the that references, I cannot collect them. You can collect from internet or so. All together, they form 10,000 taste buds. It also includes other areas besides tongues. In the epiglottis is a part of the pharynx, soft palate, under surface of the soft palate. They are also present also in the very upper part of the esophagus also. They are contained, these taste buds are there. Total 10,000 taste buds. And taste buds is important for the examination. That's short. Taste buds. Then you have to draw from your mind these structures, and book to book there is some differences in their names. Book to book you will find, and edition to edition there is a rapid changing of their names and their concept. And this is the part of physiology. They are, and also to some extent the sensory system, the pain sensations, and they are rapid changes. So in edition to edition there is much change. Researches are going on, true researches are going on elsewhere, and they are being poured in different way, and so there is differences. Suppose if you go some back editions, you will find that there are 4 types of cells. 50 to 100, they are like, they are overt structures. and they contain 4 types of cells: basal cells and type 1, 2 and 3. Type 1, 2 and 3 types of cells. 3 types of cells. And of these, type 3 cells are the mature cells, and they have their neural connection. Neural connection. They are the real test sensitive cells. But you go through the current text, you will find they differentiated in another way. They say this is same basal cells there. Basal cells there. But the other cells, they are named in this way: dark, light and intermediate. Intermediate. And they consider, according to staining reaction, and they consider these light cells, they are the 2 test cells that have neural connection. Neural connection. Previously it was thought that type 1 and type 2 cells, they are sustentacular cells. They are sustentacular cells. They are sustentacular cells. Type 3 is the 2 test cells. What is the 2 test cells? That have this innervation, they never show that this cell must be sensitive. Now, 1 and 2, in previous editions you will find this type of classification. In a test bud, they are overt-like structures, and they are 50 to 100 cells. Cells. And previously it was thought that they are the basal cells from which all other cells are derived, and type 3 cell is the most mature cells, and relatively they are sustentacular cells. But current text says there are 4 types of cells. They are basal cells from which the new test cells do generate, and they are dark, light and intermediate. And of these light cells are the most mature. And when you write something in your, there is a short note, you take it from a textbook, and if you explain it in your way, then it is okay. কারণ সঠিক কিন্তু আমরাও জানি না, ভিন্ন ভিন্ন এডিশনে ভিন্ন ভিন্ন রকম আইডিয়া আসছে। In new current edition, they means that is this, they are developing from the ultimately epithelial cells, neighboring epithelial cells, they are in close contact, and from which derived is the basal cells, and basal cells do differentiate, and they produce this type of cells. They are all ciliated cells, but this cells is mature and do have their function. It does not mean the dark and intermediate cells will ultimately not give rise to light cells. And they also think that they may be of different lineages, may not derive from a single basal cells. They may have different lineages, different basal cells they are derived. Whatever may be, they do not consider them as sustentacular cells. They are supporting cells function anatomically, but they are considered, they are all these varieties of different stages of maturation of the test cells. Whatever may be, whenever you see a test cell that bears microvilli, it is a test cell. Suppose intermediate cells or type 3 cells in this concept, and they bear, there is this nerve bending, and these are all epithelial cells, and there is a pore, test pore, and it is exposed in this way. বোঝা গেল? এই পোরটা না থাকলে কি হবে? The tongue is covered by epithelium and naturally stratified squamous type of epithelium. It may be keratinized or not, and if so, sensitive structure must come into the surface, and by means of this test pore, it comes into the surface. Surface means that have the watery part, and test producing substances, they are called testants, and they are dissolved in the watery part of the saliva, and they come in close contact with this microvilli, and they contain the appropriate receptors for them. And in taste buds, that is these are the papillae that you can recognize with your unaided eye, but they are microscopic structures, and they bear 50 to 100 cells, and they are classified in different editions of text by different way. Whatever it may be, the basic thing is that the cells that bear microvilli, and that is the basal part, is the nerve ending that merged with two test cells. Other are supporting cells or they are in the process of development, whatever it may be. They have their supporting functions. Suppose different types of cells, that is, they also microvilli, and they do not bear the nerve ending. You can say type 1, type 2 in this fashion, or in this way it is called light cells, most matching. Whatever may be the basic idea that bear some microvilli, they all bear microvilli, but the cell that is connected to the nerve is the true taste cells. And taste buds receives the fibers from 50 up to 50 afferent, more or less up to 50 afferent connections. And each nerve fiber has connection with 5 taste buds. Suppose their number is, there is this connecting in this way, there are 50 afferent connections. And each afferent connection has branches up to, that is the taste bud. Suppose taste bud 1, TB1, TB2, TB3, TB4, in this way. They branch. Each afferent fiber, they branch to supply up to 5 taste buds, and each taste bud receives up to 50 afferent types of 50 afferent types in this afferent. বোঝা গেল? প্রত্যেকটা বাংলায় বলছি, প্রত্যেকটা টেস্ট বার্ডে 50টা অ্যাফারেন্ট ফাইবার পাবে। তার মানে একটা অ্যাফারেন্ট ফাইবার যে উৎপত্তি হয়েছে, সে একটা সিঙ্গেল সেন্সরি অ্যাকশন থেকে বা ডেন্ড্রাইট থেকে তৈরি হয়েছে, সেইটা আবার আপ টু 5টা ভাগ হয়েছে, সে 5টা বার্ডকে সাপ্লাই করছে। In this way, and they have some functional implications also. And similar to that of the odorant receptors, the cells are replaced. Their half-life is 10 days or so. They are replaced. বোঝা গেল? These cells are replaced by the proliferation of the basal cells. Similarly, olfactory neurons, they are also replaced because they are coming in contact with the atmosphere, and they do degenerate, and they are supplanted by the new cells. And this is also applicable in taste buds also. They are replaced by the proliferation of the cells. বুঝা গেল? এটা দরকার আছে। Replacement, it is a peculiar thing. And this two taste producing cells that have the sensory endings, it may be 1 or in some cases it is 2, and in this case they are the 2 taste cells, and they are replaced with half-life of either by the differentiation of the cells or continuous process. If the nerve is cut, it will degenerate. But if the nerve is rejuvenated, then from the epithelial cells new taste buds will grow. বোঝা গেল? নার্ভ যদি কেটে যায় তাহলে পরে সেখানে সেটা ডিজেনারেট করে যায়। আবার যদি নার্ভটা রিজেনারেট করে তাহলে পরে আবার নেইবারিং এপিথেলিয়াল সেল থেকে ডিউ টু সাম chemical inductive influence, new taste buds do appear. Total number is roughly 10,000 together, and their number is diminished as with age. বয়সের সঙ্গে সঙ্গে টেস্ট বার্ডের সংখ্যা কমে যায়। যেমন আমি যখন এর জন্য চা খাই, আমি একটু চিনি বেশি দিতে বলি, নাহলে পরে আমার স্বাদ কি হয় না। বুঝতে পেরেছো? এর জন্য আমাকে একটু বেশি করে চিনি দেয়, ওরা যে জানে যে এই ভদ্রলোকের বয়স হয়ে গেছে, চিনি বেশি দেয়, কারণ আমার টেস্ট বারের সংখ্যা কমে গেছে, আমার অনুভূতিটা একটু কমে গেছে। বয়সের সঙ্গে সঙ্গে বুদ্ধি শুদ্ধি সবই লোপ পায়, হ্যাঁ, কিন্তু হায়ার ফাংশনটা দেখা যায় অতটা লোপ পায় না, কারণ সেখানে অভিজ্ঞতা চর্চা সবসময় হচ্ছে, কিছুটা কমে যায়। এই জন্য আমাদের মতো লোকদের ঠকাতে লোকের সুবিধা হয়, দোকান বাজারে সবসময় আমাকে ঠকায়, আমি লোককে বলি তুমি ঠকাতে পারছো না, তুই কি হবে, তোর পেছনে থেকে কি হবে, হ্যাঁ। এই জন্য সেই টেস্ট ছিল সংখ্যা, কেন বললাম তার মনে থাকবে। যেমন ওলফ্যাকশনের সঙ্গে এজেড ইন্ডিভিজুয়ালের ওলফ্যাকশনের ক্ষমতা কমে যায়, বেশি সেন্ট লাগে। আমি সেন্ট যখন দিই একটু বেশি করে দিই, লোকে বলে যে আপনি এত সেন্ট দিয়েছেন কেন, কারণ আমার তো ঘ্রাণ শক্তি কমে গেছে। এগুলো ফিজিওলজির ব্যাপারটা তোমার জানতে হবে, প্যাথোলজি নাই জানলে। Now it is the almost all the test, but now the innervation is important for the examination. তোমাদের টেস্ট আমি তোমাদের দেখিয়ে দিয়েছি কি করে করতে হয়, যারা আমার কাছে গিয়েছিল কিভাবে টেস্ট করতে হয়, টাং প্রলং বাইর করতে হয়, এবং সেই অবস্থাতেই বাই মিনস অফ এনি কার্ড অর উইথ দি প্রোট্রুডেড টাং you must give this, otherwise when tongue is inside so it will spread from one side to another and from front to back, so different types of nerves and different sides of the nerve are involved, so we have to keep the tongue protruded, বুঝতে পেরেছো, জিভটা প্রলং করে রাখতে হবে, এইভাবে তোমার দেখতে হবে। জিভ যে ভিতরে নেবো, একটা সাইড থেকে অন্য সাইডে চলে যাচ্ছে, সামনে থেকে পিছনে চলে যাচ্ছে, ডিফারেন্ট নার্ভ, একই নার্ভের দুটো সাইডের মধ্যে কনফিউজ হতে পারে। এই জন্য এটা হচ্ছে ক্রাক্স অফ দিস টেস্ট। এটা যারা আমার কাছে প্র্যাকটিক্যাল দিয়েছিল, আমি তাদেরকে তোমার দিয়েছি না, তোমাদের দেখিয়ে দিয়েছি না, হ্যাঁ, তখন হেসেছিল, কিন্তু সেটা মাথার মধ্যে এদের ঢুকে গেছে, বুঝতে পেরেছো? এভাবে টেস্টটা করতে হয়। এখন নার্ভের ক্ষেত্রে তোমার হচ্ছে যে এখানে অ্যান্টেরিয়র 2/3 অফ দি টাং, এখানে যেখানে, এবং সেখানে হচ্ছে তোমার কটা টিম্পানি ব্রাঞ্চ, লিঙ্গুয়াল নার্ভ, quarter tympani branch unites with the lingual nerve, and lingual nerve, so quarter tympani branch, facial nerve, facial quarter tympani branch of the facial nerve, or it also travels through greater superficial petrosal nerve in some cases, greater superficial petrosal nerve, and posterior 1/3 of the tongue, including this valet papilla, and the glossopharyngeal, it is the glossopharyngeal nerve, 9. And epiglottis and most other structures, they are carried by the vagus nerve, epiglottis, and there is epiglottis, they are mainly this the vagus nerve. Soft palate, under soft palate, it may be carried by the facial nerve or by the vagus nerve. This is this, and for details you have to consult the anatomy texts, Gray's Anatomy also for details. But roughly it is asked in the examination in physiology. Details of anatomy we are not interested. Now their destination is that they enter suppose it is the midline, and then from the taste buds, there is the facial nerve, they enter into the geniculate ganglion, mainly facial ganglion or geniculate ganglion, and then it is the, suppose it is this 9th nerve, petrosal ganglion, and this knife, and from the 10th by nodose ganglion, and they enter into the, on that side of the dorsal part of the nucleus of the tractus solitarius, and they then here the second order neuron do appear, and they will travel on the ipsilateral side. If you go to back editions, you will see that the fibers are crossing the other side. All the text, older edition. But now they are in the ipsilateral on the same side. And it goes to the, and it is mixed with, there is the medial lemniscus or fillet, medial lemniscus. Into thalamus, in the thalamus, they terminate in the thalamus. Along with other fibers, touch pain or fibers in the ventral posteromedial nucleus. And then by the thalamic radiation, they enter into this termination in the cerebral cortex, in the foot of the postcentral gyrus, in the insula, and the some part of the anterior part of operculum, frontal operculum. In three areas, foot of this postcentral gyrus, and also the insula and the and nearby part of the frontal operculum. This is the area of termination in the cerebral cortex, from thalamus to cerebral cortex. Now this modality of the taste sensation, what are the modalities of the taste sensation? Modalities of the taste sensations are there are basically five types. Older taste, fourth sensation, and then umami is added, and they also mention that there may be other varieties of taste, primary taste, but commonly five types they do consider. That is the sweet, salt, sour, bitter, amino acids or umami, Form of, and these are the five types, classical, they are appropriate receptors. Previously it was thought that from this part, in this tongue, the anterior part on either side, it is related to, that is the sweet part. Then it is the, to some extent, sweet, salt, sour, and this part is bitter. And the middle part of the dorsum, they do not have. But if this test is birds, they are in the papillae, but does not mean they are do not place without papillae, and they consider that that central part has no taste sensation. But in reality, so such classification is relatively crude. There may be some anterior part is more sensitive to sweet. Does not mean the sweet sense cannot be elicited from other areas, and it is applicable to all others. And there was much confusion in this. If we have time and we go to different editions of text written by different authors, then you will be in a confused state. But this confusion has to some extent eliminated with the development of the knowledge. That is, when the receptors has been identified, then it has been possible that that is this cloudiness of knowledge in the previous days are to some extent dissipated, and the receptors have been identified, and their mode of action, molecular mode of action is known for a good number of cases, and it is progressive. In addition to text, reliable text, you will see changes, or at least on this part. What are the receptors, modality of the test, what are the receptors, and their mechanism of action, how the receptors do have their effect. Do have their effect. And it has been seen that there are some test receptor cells, they can respond to different modalities. Different modalities. Because they can harbor different types of receptors for the different modalities. Or in some cases, a test cell may be particular. It will produce only that receptor. That receptor. That receptor. So, it is to some extent to that of the sensation of light also. Normally, light sensation is due to mixing of the monochromatic lights, different monochromatic lights, and the taste sensation we are getting, and it is a mixed type of sensation. And very little work can be done in their higher centers, cerebral cortex or so, and then so the knowledge is to some extent incomplete. But to some extent, real picture is becoming clear. That is, there are some receptor cells, they are relatively subpolymodal in this case. Some may have SAVA receptor and suppose a beta receptor. Some may contain all four and some may be specialized for particular modality. Some modality or the modality of the test sensation. And similarly, they supply the higher functions and the brain can judge them. So location is not so much important. And this type of location, I may commit some mistake or so. You will find it is older classification that anteriorly it is sweet, posteriorly it is bitter, and then this salt sour at the one is anterior and one is to some extent medial and to some extent lateral. And I may confuse whatever it may be, but these concepts are almost gone, and all the areas are sensitive to particular. But in reality, it is the sweet taste is better appreciated from this. There is the apex of the tongue, and bitter to some extent it depends upon the concentration of the receptors. Because the violet papillae, they contain good number of receptors, so bitter taste can be considered, and it is the behavior of their mode of interaction. And the molecular mechanism is like that in case of salt sensation, and in that case the receptors is this receptor, it is a, suppose it is a, in this type of receptor they are, and these receptors they are sensitive to salt and sodium chloride, then the sodium ion can do enter through this, and this picture should be better. You take a single subunit, and when the sodium ion do enter, it causes depolarization, and depolarization when reaches a limit, then the action potential can be generated. Even action potential can be generated with the test cells also, but commonly it is not so. Depolarization is sufficient. Whenever it is depolarized, then they liberate some transmitters. It may be serotonin or so, and textbooks is not clear about this. And then action potential can generate. So generator potential is produced by the entry of the sodium ion in this case. Sodium ion. And it is known by there is ENA, epithelial sodium channels. Epithelial sodium channels, ENS. You are familiar with the name of the ENSC? Regarding the function of the mineralocorticoid and the distal part of the nephron, same ENSC. And it is the same compound that inhibits it, amyloid. Amyloid inhibitable sodium, amyloid. Can inhibit this. But this inhibition is not complete. So that other types of channels or so, sodium channels may be responsible. ENSC, epithelial sodium channels, by means of which these are entering. In the text you will find this type of some graphic pictures. The two subunits through which sodium is entering. And each subunit, there is the carboxyl end and N-terminal end is within the cytosine. But for ease of application, description, I described in this way. Now in case of, but amyloid when is applied, this sensation is reduced but not abolished. So it indicates that other types of sodium channels may be there or cation channels may be there. Now in case of Sauer test, it is the hydrogen ion that is primarily responsive. Hydrogen ion. And in this case, it is also ENS. Hydrogen ion enters, hydrogen ion enters. And this hydrogen when enters, it may have some action on the potassium channels. And potassium exit is when it is reduced, potassium channels. And then what occurs? It is more depolarized, and the action potential may start in the afferent node. It is the same ENSC or it is HCN type. Hyperpolarization induced cyclic nucleotide gated channel. Whatever may be, it is present here also. That may be responsible. Hyperpolarization induced cyclic nucleotide gated type of channels, cation channels, and that may be responsible. And to some extent by the ENS that permits not only sodium ion but also hydrogen ion. And this hydrogen ion, this concentration of hydrogen must be there, but it does not mean this is pH. Threshold of that is a hydrochloric acid sensation is 100 mmol per liter. 100 mmol per liter. But if it is organic acids at a much higher pH, they are more acidic. It is there as their organic compounds, their capacity of penetration is more than this. To the epithelial cells, whatever may be there is a test pore also, then their rate of penetration is more. So citric acid having a pH more than this, and they are more active in generating the Sauer sensation. It is due to their greater penetrating ability. That is written in the text. And in this case, this 100 mmol per liter is the, in this case it is pH is equal to which value? 100 mmol per liter যদি হয় তাহলে pH কত হবে? 100 mmol per liter যদি হয় তাহলে pH কত হবে? বার করো তো। হ্যাঁ, pH 4 হবে। কাজে সেক্ষেত্রে এই ক্ষেত্রে তার মানে এটা রিলেটিভলি স্ট্রং অ্যাসিড না হলে হবে না। হ্যাঁ, কিন্তু organic compounds much higher pH is also better. Now these other types, that is the sweet, bitter and amino acid or umami, they are all G-coupled receptors. They are all G-coupled receptors. GPCR. G-coupled receptors. Suppose in case of sweet, and they are G-coupled receptors. Suppose it is cell membrane. G-coupled receptors, and they unite with the G proteins, heterotrimeric G proteins. This is the alpha, beta, gamma in this cell membrane. There, and they are of different families, and they are heterodimers. T1 varieties. T1 varieties. T1 and this is the T2. T2. This type of, there are G protein coupled receptors and they are heterodimers, and in this way they form a heterodimer and this and up to this, and they activate this when this bind to this receptor, suppose sweet molecule, they are when uniting with this, then there is activation of this G protein coupled receptors, G protein, and this alpha unit becomes separate, this alpha unit, and then it will unite with the PLC, phospholipase C beta 2 type of receptor, and it increases the IP3, and they increase the calcium transport in the calcium channel, calcium increase this. In this way, they have their effect. T1. T1 variety. Test one variety. T1 R2, T1 R3. And when they becomes activated with these appropriate ligands, sweet substances, they have their effect in this way. Like G coupled receptors, they are all 7 transmembrane domains, serpentine receptors, and this then is alpha unit becomes separated, binding with GTP, replaced by, in this case it is bind to GDP, and when GTP activated, then the alpha unit with the GTP, and it will activate the phospholipase C beta 2 variety, and IP3 is produced, inositol triphosphate, and it causes somehow entry of calcium onto these calcium channels, and this calcium entry is responsible for depolarization and the liberation of the neurotransmitter, Even the test cells can generate action potential. So they are called, in some tests they are called neurons, similar to that of the olfactory, but their term paraneurons are used. They are not neuron proper, and they are called, you may call it the paraneurons. Commonly they generate, there is the potential, and due to this, that is, and potential is sufficient for liberation, and when then this is sufficient, then calcium ion do enter, and when the depolarization is sufficient, then there is a liberation of neurotransmitters, and they are carried by the afferent nerves with it, afferent nerves linked with it, afferent nerves. With the action potential can be produced, action potential in this way. Now, in case of, there is the amino acids. Suppose umami, amino acids. In the amino acids, monosodium glutamate or some nucleotide also, they are responsible, and it is T1R1 amino acid. Amino acid T1R1. This remains constant. And the basic mechanism is the same. Monosodium glutamate or other some nucleotide also responsible for them. And the basic mechanism is the same. This heterodimer is T1R1 and T1 R3 and it is equal for them. তোমরা বই থেকে দেখে নিও, আমার যদি কোনো ভুল হয়, ভুল হতে পারে, কারণ আমি তো দেখে দেখে পড়ছি না, কাজে এটা ভুল হতে পারে। কাজে তোমার একটা বেসিক কনসেপ্টটা that is the required. They form heterodimer and these other mechanisms are the same. G protein covalent receptor GPCR and then there is activation of phospholipase C. Phospholipase C acts upon the membrane phospholipids. Ultimately they derive diacylglycerol and inositol triphosphate. Here inositol triphosphate is important and that causes activation of this some calcium channels and these calcium channels calcium do enter and do have their effect. Depolarization along with this liberation of neurotransmitter in this way. But in the bitter taste, it is almost the same, but in this case it is the bitter and it is the T2R variety, taste 2R variety receptor variety. It is cell membrane and it is, suppose it is a solitary structure and there are separate G proteins alpha, beta and gamma, whatever it may be, and but this they are gasducins. They are termed GUSTDUCIN, G proteins, gasducins, and their behavior is similar to this. But this G protein is different from this. To some extent, structurally different from this. It has been proved by different very intelligent type of experiments. It is separate from this, but the basic pattern is the same. That is, this activation of PLC beta 2, IP3, and this part is same. But here this G protein is to some extent different. And what is the reason is not known, but we are much sensitive to bitter taste. Suppose in hydrochloric acid, we mention it is 100 mmol per liter. But in strychnine, it is 1.6 mmol per liter. In strychnine. And in case of quinine, is to some extent more. It may be query, that is the 8 mmol per liter. You will confirm in your text. Quinine. And quinine has the property of entering into, they have their effect without avoiding this type of receptors. In older text, they mean they had direct effect upon the G, that is this type of receptors. But present day mention that they can avoid these receptors and they can enter into the cells and they may have their effect by some potassium sensitive cells. There is the potassium channel blockers, or they may have simultaneous effect by various routes. And the idea is that it is the most of the natural poisons are bitter taste, alkaloids, they are bitter taste, and so their threshold value is also low. It is strychnine and quinine is. So there are multiple effects. Similarly, in case of that is the amino acids, monosodium glutamate, only this type of, they have also inotropic receptors, and it is the metabotropic receptors. And why they should not have they have their inotropic receptors. Similarly, inotropic receptors are also there in case of this type of umami taste. Inotropic receptors, they have direct effect on the ion channels, like acetylcholine nicotinic receptors, inotropic receptors, they also. So there are, and everything has not become apparent, but to some extent is going to be clear. If you read older text, it becomes very difficult to understand or retain in the memory by common intelligence. There is intelligence of a low variety like me to grasp everything. But as days progresses and পেইন পাথওয়ে, হ্যাঁ, তাতে তোমার টাইম আছে, এরপর তোমার অ্যানাটমি কাজটা বোধ হবে না। দেখ আমার যতটুকু জ্ঞান বুদ্ধি আছে সেটা আমি তোমাদের বলে দিচ্ছি। হ্যাঁ, সবটাই জানবে যে আমি যেটা বলছি সেটা সবসময় ইনএডিকুয়েট, কাজে ইউ হ্যাভ টু সাপ্লান্ট ইউর নলেজ। আমার জ্ঞান খুব সীমিত, কাজে তোমরা এটা আবার নিজেদের সুপিরিয়র ইন্টেলিজেন্স দিয়ে সেটা ভিন্ন ভিন্ন বই থেকে, ইন্টারনেট থেকে জ্ঞান সংগ্রহ করে নেবে। এবং এটাই আমাদের ইচ্ছা। কারণ এখানে তার মানে এখানে এই অবধি মোটামুটি তোমাদের আলোচনা হলো। হ্যাঁ, প্রায় আমি কমপ্লিট করে দিয়েছি। আর অন্য যেগুলো আছে বই থেকে দেখে নিতে পারবে। আর পেইন সেন্সেশন, that is this, and it is also changing quickly. Pain sensation. Pain is produced by any stimulus that causes damage or to some extent temporary harm to the tissues. And it is a protective device. Pain is a protective device. And the pain receptors are the free nerve endings. Free nerve endings. It is more present in the superficial part of the body than in the deeper structure. And there are two types of pain. One is fast pain, another is the slow pain. Fast pain or slow pain, or first pain or second pain in this. Fast pain is produced by the A delta fibers, and slow pain is by the C fibers. A delta fibers are thinly myelinated. A delta fibers. A delta fibers are thinly myelinated, 2 to 5 mm in diameter. Conduction velocity is 12 to 30 meter per second. Conduction velocity. And C fibers are non-myelinated. Non-myelinated. 0.4 to 1.2 mm. And conduction velocity is 2 meter, approximately 2 meter per second or so. Slow process. Slow process. And unmyelinated. And it is thinly myelinated. And the first pain that it produces whenever you prick. Whenever you prick, in the practical also, you feel initially a sharp, bright, sharp localized pain. You can easily locate this localized pain. But after some time, then that sharp pain will disappear quickly. But after that, you are feeling some type of pain, depending upon the pressure. It may be intense also, but it is dull, diffuse, not and poorly localized. So, and as the distance of application from the brain increases, their temporal separation is there. বুঝতে পেরেছো কি বলতে চাইলাম? The distance of the brain is increased, their temporal separation is better evident. যেমন তোমার বিগ টোতে যদি একটা প্রিক করি, তাহলে দুটোর মধ্যে যে টাইমের ডিফারেন্সটা, আর যদি নেকে প্রিক করি তাহলে দুটোর মধ্যে টাইমের ডিফারেন্সটা বুঝতে পারবো। যদি আমরা যে টোতে প্রিক করি তাহলে পরে দেখা যাবে টাইম ডিফারেন্সটা তুমি পরিষ্কার বুঝতে পারছো। হ্যাঁ, কারণ অনেকটা দূর থেকে সেটা আসছে, স্পিড কারো কম বেশি, এই জন্য যত সময় যাবে, যত দূরত্ব যাবে, তত সেটা এভিডেন্ট হবে দুটো আলাদা। কাছাকাছি থাকলে পরে টেম্পোরাল সেপারেশনটা কম হবে, এবং এটা দিয়েও বোঝা যায় যে এটা সেপারেট ফাইবার সিস্টেম দিয়ে আসছে, সেপারেট ফাইবার সিস্টেম দিয়ে আসছে। এবং এই ক্ষেত্রে বেশিরভাগ ক্ষেত্রে পেইনটা ক্রস করে অপোজিট সাইডে যাচ্ছে, অপোজিট সাইডে যাচ্ছে, অপোজিট সাইডে যাচ্ছে। Suppose in this, if you consider this, এরকম হয়ে যাচ্ছে। এটা হচ্ছে পেরিফেরাল, এটা বডি সারফেস থেকে আসে, হয়তো স্কিন থেকে আসে। ডরসাল রুট গ্যাংলিয়ন, this ganglion, and it is the dorsal root cell, it is also called, and it is the projection neuron or dorsal horn neuron. It is also called dorsal horn neuron, and it is a dorsal root neuron, and it is processed to the opposite side, and anterolateral system, spinothalamic tract, or anterolateral system, ALS. Better is anterolateral system. Ventral and this lateral tract, spinothalamic tract, this system, and it will go to the brain. Now this termination, suppose in case of fine touch, vibration, dorsal column lemniscal system, and in this case the initial part is uncrossed, and they have their neurons in the and they cross at the level of the lower part of the medulla, fasciculus gracilis into gracile tubercles and fasciculus cuneate tubercles, and from which postganglionic fibers arise, and they from medial lemniscus, and they ultimately terminate level line structure in the ventral posterolateral nucleus of thalamus, from which the thalamic radiation do arise, and they have their ultimate destinations in the S1 and to some extent S2, postcental gyrus, in the inverted fashion, S1 and S2, that is in the roof of the sylvian fission, where relatively crude distribution is there. And in this case, they do follow to some extent this route, but they terminate at the level of the spinal cord and the brainstem in the reticular formation. They may go even to the thalamus, non-specific projection nuclei, centromedian nucleus also. And in this way, they causes arousal phenomenon. ব্যথা পেলে পরে সঙ্গে ঘুম ভেঙে যায়, arousal phenomenon, reticular formation, directly or indirectly have their effect by the thalamus, or even directly to the cerebral cortex, mostly through the thalamus. Different, not specific, they have their arousal phenomenon. They may also trace to the hypothalamus also. And PT, PET, positron emission technique, and MRI, magnetic resonance imaging techniques, and these are the techniques that has been done in the individuals to show which part of the brain is affected by pain sensation. They are S1, S2, and also other areas. Even cerebellum also to some extent involved. Prominently, it is in the S1 and S2. And they have distribution in the hypothalamus, thalamus, reticular formation, brainstem in this end. And the better perception, that is A delta fibers, that is the sharp localized sensations, and they are relatively new, he has mentioned, and there are older paleo, there is older to some extent, the other locations. And the neurotransmitters are normally, that is first pain by the glutamate receptors, liberated neurotransmitter, NMDA, through which it has been, and the slow pain mainly by the substance P and calcitonin gene related peptide, and their termination in the dorsal horn to some extent different. And you have read in every text, they are laminated structures. In the spinal cord, they are laminated, and C fibers terminate in 1 and 2 layers, and the A delta fibers say 1 and 5 layers. But if you consult the current anatomy text also, they are much more complicated. This is to some extent oversimplification in physiology texts. Understanding is better required. And then there are two types of pain. One is the acute pain, another is the chronic pain. পরে কোনো ক্লাস নেই তো, acute pain and chronic pain. Acute pain you have tested in the laboratory. When you biopsy pin prick, you feel a sharp, bright, localized pain. With eyes closed, you can easily identify the place. But after that, there is some dull, intense, diffuse pain, poorly localized, and that is due to these C types of fibers. And this physiologic and pathologic conditions, they also, in this case, it is to some extent there is a physiologic pain, and pathologic pain are of two varieties. One is neuropathic pain, and another is inflammatory pain. আমাদের সবার অভিজ্ঞতা আছে, কারো কোনো খোসপাচার বা ফোড়া হয়নি এরকম লোক বিরল। এটা তোমাদের নিশ্চিত কারো ফোড়া হয়েছে, যখন ফোড়া হবে তখন দেখবে সেই জায়গাটা হাইপার অ্যালজেসিয়া পাওয়া যায়, সামান্য ছুলেই প্রচন্ড ব্যথা লাগে, ইভেন টাচ করলেই ব্যথা লাগছে। When it is due to relatively milder stimulus that normally ignored, and that is called hyperalgesia. And when it is done by the other stimuli than pain, it is called allodynia. Hyperalgesia and allodynia. Allodyne. Mild stimulation, if it is this to some extent, it is the pain sensation, eliciting pain sensation. If it is very small, you can ignore. But if there is some inflammation nearby, then chubina. And when you touch by a cotton wool, then there may be pain sensation. And it is called allodyne. Hyperalgesia and allodynia. And it is seen in case of inflammatory pain. Besides this, there are some chronic pains. There is neuropathic pains due to damage of the nerves. Chronic pain. In this case, there is no inflammation, but there is pain, and it may not be properly poorly. It may be poorly localized, and it may be even continuous also. There is a term causalgia. Continuous pain. After some trauma, there is healing of the other tissues, but the pain may continue. And it may be to some extent continuous process. Continuous pain is there. And is very much distressed. And due to my pain, osteoarthritis, I am myself feeling this. And it is a deep tissue, not poorly localized, and it has some autonomic effects. When the pain is, then there is sweating, there may be fluctuation of the pressure, and if it involves viscera also, then there may be vomiting also, nausea, vomiting also. I have the type of feeling, but it is not continuous, to some extent remittent. With rest, it remits. And when you observe me, that after going to some distance, I am standing, I am taking rest, and then after few steps, I am again, and in this way, I am somehow reaching my destination. And this pain is poorly localized. It is not possible to particularly localize that sensation. Deep pain. In the deep tissues, there are less number of A delta fibers, so it is difficult to find the localization. But the pain is severe. Now, in case of causalgia, it may be possible if there is nerve damage, then there is continuous pain. Very difficult to treat. And there is hyperalgesia, allodynia, along with some sympathetic, some effect on sympathetic nervous system on this part. Chronically, there may be skin may be thin, shiny, and even hair growth. And here the continuous pain is due to the liberation of neurotransmitter from the sympathetic system. And how it is possible? As the growth occurs. Anatomy, we have to consider this. Suppose in this case, if you consider this, in this case, that is the, suppose the, it is the, that is the, if it is the lateral horn, and it is in this case, this, it will come back, it is come to, and then it is white rami communicantes, grey rami communicantes, and white rami communicantes. Beside this, it has also some afferent fibers that have their dorsal horn neurons also. And whenever you see, then there is, and whenever, if you consider the motor part, in the lateral horn, that harbors the preganglion neuron, and by means of this, there is grey rami communicantes, excuse me, it is the white rami communicantes, it is coming to, and this grey rami communicantes to this postganglion neuron, and they are traveling along with the peripheral nerve. And it is myelinated, white rami communicantes, it is the grey rami communicantes. And whenever suppose there is some section in this case, then it is possible that sprout will develop, and this part will degenerate, because this is the nucleated part, it remains close to the nucleus, and then this twig develop. Suppose this twig develop, it will travel and it will directly have its effect. Directly have its effect. Whenever the nerve is cupped, the peripheral part of the sympathetic nerve and all other nerves will disappear, degenerate. But those connection with the nucleus, they will remain alive. And they will try to, similar to this nerve section, and they will give sprout. And these sprouts, they can appear and they may have their connection with the dorsal neuron to the nociceptors. And there is continuous liberation of norepinephrine. Norepinephrine. Clear? Here there is liberation of acetylcholine. But here is norepinephrine. And it is the physiological or anatomical basis of continuation of the pain. Due to there is constant activity. Sympathetic nervous system is continuously active, because we are, till our death, we are always in some form of stress, and they are continuously active. And there is continuous liberation of norepinephrine. And norepinephrine, when it supplies this, there is continuous stimulation of this pain path. And so there may be exacerbations, or there may be some lessening, but the pain will be continuous. And treatment is by alpha adrenergic receptors, alpha 1 adrenergic receptors, blockers, alpha adrenergic receptor blocker. But it is not known exactly why this is more important than beta adrenergic blocker. Probably it is this type of receptors that do grow. Yeah. In the inflammatory type, there is the liberation of substances at the localized area. It is called inflammatory soup, and it contains the nerve growth factor along with other factors. In the text, they describe in this way. Suppose in inflammatory pain, now in this connection, you have to know also how this allodynia should develop. Suppose touch receptor fiber with different color, allodynia, how it develops. Suppose it is a touch receptor, and it is periphery, and it is entering, and they are traveling in the same direction, in the same direction, in the ipsilateral side to terminate. Suppose it is the dorsal tooth ganglion carrying a fine touch, and it will not cross, and it will travel to the same side as dorsal column lemniscal system. And what occurs? In chronic cases, then there may be some branch may develop, to some extent gate control theory, but in that case, it will have this effect. It will have this, and in this condition, it will have its effect. It may be give rise to some branch. It directly or indirectly may have this effect upon this. Further development, due to this further development of this twig, so that whenever this from the skin there is the fine touch is present, it will activate this. It will activate this. So it is the pathway of allodynia. Normally, dorsal column lemniscal system in health, they have their effect, relatively negative effect, gate control theory. And in that case, there is an interneuron that is present. In physiological conditions, there is an interneuron that is present. Interneuron. And this interneuron will liberate some enkephalin-like substance, inhibitory substance. They can inhibit. It is the physiologic process. But in case of chronic cases, it is possible that there is twig that directly stimulates this. So that this, whenever this fine touch is applied, কোন একটা জায়গায় যদি একটা ফোড়া হয় বা কোন একটা জায়গায় যদি ঘা হয়, তুমি দেখবে সামান্য তুলো দিয়ে টাচ করলেও সে কেঁপে উঠছে, প্রচন্ড ব্যথা হচ্ছে। তাহলে অ্যালোডাইনিয়াটা ডেভেলপ করলো কি করে? How the allodynia do developing? There may be twig directly from this, that is the thick fibers going. That is the dorsal column lemniscal system pathway, from which a twig may go to this pathway. In this way, it is given in different texts. আমার যতটুকু জ্ঞান বুদ্ধি আছে আমি তোমায় কুলিয়ে নিলাম, তোমরা আবার এটা বিচার করে দেখবে যে আমি ভুল বলেছি কি না কি বলছি। হ্যাঁ, it is enlarge your process of thinking, how it is possible. ডরসাল কলাম থেকে নরমালি গেট কন্ট্রোল থিওরি আছে। যেমন কোন একটা জায়গায় চোট পেলে পরেই দেখবে আমরা নাড়াই, অথবা ফাইন টাচ করলে পরে ব্যথাটা কিছুটা কমে। ইট ইজ এ ফিজিওলজিক প্রসেস। এবং সেক্ষেত্রে যেহেতু নসিসেপ্টরটাও তোমার সমস্ত স্টিমুলেটরি পার্ট, আর টাচটাও স্টিমুলেটরি পার্ট, কাজে স্টিমুলেশনকে ইনহিবিশন করার জন্য একটা ইন্টারমিডিয়েট নিউরন দরকার। সেই নিউরনটার সেক্ষেত্রে আইদার এনকেফালিন বা কোনো ইনহিবিটিং সাবস্টেন্স লিবারেট করছে। দ্যাট ইজ দি ফিজিওলজিক প্রসেস। বাট ইন সাম কেসেস ইট ইজ পসিবল that whenever there is allodynia, then you can see it is a chronic process due to some effect of some nerve growth factor or so. A twig can develop directly up to this, from this touch sensitive pathway to the pain sensitive pathway. So whenever there is fine touch, then there is pain sensation at that particular localized area. এবার বোঝা গেছে জিনিসটা? Now the inflammatory soup, they contain different substances. Suppose in the good test, they will the part of the skin, and here there is some trauma. And then in this condition, you will find that this is now, suppose it is the termination, it is the free nerve endings in the tissue, and there is the damage at that site. And there is good numbers of substances will be available there. Different types of inflammatory cells, they will liberate different substances. Suppose it has a one twig that is liberating, suppose calcitonin gene related peptide and substance P, that is the substance P, and they will have their effect upon some cells, macrophage cells, monocyte cells, macrophage or other immune cells, and they are liberating histamine. 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 And histamine will potentiate these receptors. Besides this, there may be liberation of bradykinin. And from platelet, you have get serotonin. You are getting serotonin. From platelets, whenever there is trauma, there is destruction of the blood vessels, there is liberation of cells, including the platelets. They are ruptured and they will provide the serotonin, and other substances. There will be produced like prostaglandins, including the neurons. They will produce. And potassium ion is available. Potassium ion. And potassium ion, suppose whenever the potassium level at any body site increases, extracellularly, then there is depolarization. And whenever there is depolarization, the nerve cells will be more sensitive. Prostaglandins do have that effect by G-coupled receptors. Prostaglandins can be produced by the neighboring cells or also by the neurons itself. They also divide. And in this way, they are mutually they are secreted. They also contain nerve growth factor. And they contain this. And this substance P, and it will have that effect upon the blood vessels. Substance P will help in exudation of fluids between the endothelial cells. And there is vasodilatation with the help of calcitonin gene related peptide. Vasodilatation. And so there is edema will develop. যখন কোনো চোট লাগলে দেখবে জায়গাটা ফুলে উঠেছে, এটাকে ঘুষি মারলে দেখবে মুখটা ফুলে উঠেছে খানিকক্ষণ বাদে। তখন তোমার ওইটুকু জানলে তোমার হবে না, ঘুষি মারবে কিন্তু কেন জেলে উঠেছে তোমার সেখানে কমপ্লিট অভিজ্ঞতা চাই। এর জন্য বুঝতে পারছি তো, অজ্ঞানের সঙ্গে জ্ঞানেরও সেখানে একটা সমন্বয় চাই। তারপরে এগুলো ক্যালসিটোনিন জিন রিলেটেড পেপটাইড, এগুলো তুমি ডিটেইল দেখে নেবে। হ্যাঁ, এগুলোর জন্য সব আমার মুখস্থ রাখার কথা নয়, আমার অত বিদ্যা বুদ্ধিও নেই। আমি তোমাদের শুধু আইডিয়াটা ধরিয়ে দিতে পারি। আমার কথা বুঝতে পেরেছো? দেন ইউ উইথ ইউর সুপিরিয়র নলেজ ইউ ক্যান বিল্ড দ্য টাওয়ার অফ ইউর নলেজ। এটা বুঝতে পারলে যে এটা কিভাবে হলো এই সুপগুলো, and they have their effect upon this. And nerve growth factor, they do have their effect. Nerve growth factor, in chronic cases they can change or they change the character of the neuron. Even this touch receptor pathway can be converted into pain receptor pathway. Due to effect on the higher centers, they can be converted. এবং এটা ক্ষমতা নার্ভ গ্রোথ ফ্যাক্টরের আছে, কারণ তার যেমন ড্যামেজ নিউরনকে রিপেয়ার করার ক্ষমতা আছে, তার আবার চরিত্র পাল্টাবার। It can have effect upon the touch receptor pathway. This touch receptor pathway can be converted into a pain pathway. It has the capacity. এবং it is proved. That is the touch receptors, they can be converted into pain receptors. Because not only at the early part, but at the central part, it may produce. Nerve group, they have this effect. They can rejuvenate a damaged neuron. Similarly, they can act upon a neuron when it is in a continuous process of stimulating, and it may convert its character. A touch sensitive pathway may be converted into a pain sensitive pathway, because different types of proteins will be produced, and their connection will be at the higher level somehow in the different. Everything in this subject is not known, and addition to addition, there is some addition and alteration, and it is possible that there is change in their character, So it is this type of, and they contribute to this excessive pain. Now regarding the opioids, there is few minutes of time. There is regarding the opioids, you have to know how do they operate. Endogenous, that is the pain inhibiting mechanism. how they operate. Suppose in this case, if you consider this, then there is this opioid receptors. Opioids means that is the opium-like substance. Opium-like substance. And these opioids, that is the simplified structures. They have a good number of amino acids and they all contain either met-enkephalin or leu-enkephalin. Methionine enkephalin and leucine enkephalin. The basic structure is that there is this tyrosine, glycine, phenylalanine and methionine. In case of met-enkephalin. And the structures are same and except there is leucine. Tyrosine, glycine, phenylalanine, and these are the met-enkephalin, and they are active. They may be associated with other amino acids. And they are called opioids. And say they bind to opioid receptors. এই জন্য ওপিয়ামের জন্য সে আলাদার জন্য রিসেপ্টর তৈরি করতে যাবে না। তার মানে দেহে কিছু জিনিস উৎপন্ন হচ্ছে যেটা পেইন ইনহিবিটিং কাজ করছে, এন্ডোজেনাস পেইন ইনহিবিটিং সাবস্টেন্স। ইট ইজ কল্ড এন্ডোরফিন। এন্ডোজেনাস মরফিন থেকে এন্ডোরফিন। এর মধ্যে হচ্ছে কমন স্ট্রাকচার ইজ দ্যাট দে মেড এনকেফালিন and leu-enkephalin. And they are different types of precursors. Pre-pro-enkephalin. It is produced in the brain and also in the adrenal medulla that secretes adrenaline. They produce this. And they are present in the brain and they are basically neurotransmitters. But they are also present in the blood. But their effects in the circulation is not known because they cannot easily penetrate the blood-brain barrier. বুঝা গেছে? আর এটা হচ্ছে প্রোডাইনরফিন। ডিফারেন্ট টাইপ জায়গা ইন্টেস্টিনে তৈরি হয়। ডাইনরফিন 17, ডাইনরফিন 1 to 8. They are present in the duodenum and elsewhere. And pro-opiomelanocortin. Pro-opio are familiar with the pro-opiomelanocortin. Intermediate lobe of the pituitary, anterior pituitary. They are the precursors of different substances like ACTH, beta endorphin, and also melanocyte stimulating hormones. In adult individuals, their role is limited. ACTH has its main role in stimulating the melanocytes, melanin formation. So in Addison's disease, whenever there is less glucocorticoid, more ACTH is liberated, and it act upon the melanocytes of the skin and elsewhere, and there is causes pigmentation, hyperpigmentation. Melanocyte stimulating hormone in the adult individual, in human beings, are less important, and very small amount can be produced. Its function is taken over by ACTH because it has some amino acid composition similar to that of the ACTH, and they have their effect upon the melanocytes. It is the precursors, pre-proform, proform, and these opioid receptors, and these opioid receptors are generated by the cells. Opioid receptors, and they are these receptors, opioid receptors, OR is produced at that part, OR, and also at higher level, in some cases, opioid receptors are produced. It is produced in this part and it is transported in this way. Opioid receptors are in the periphery and in the central part, opioid receptor. In the periphery, there is generation of the opioid peptide-like structures. Opioid peptide structures in the inflammatory fluid, opioids. And these opioids do have their effect. Opioids do have their effect. And similarly, opioids do have their effect in these opioids. And these opioids, in this case, is coming from the neurons, some intermediate neurons. And fibers coming from above. They are liberating serotonin, 5-hydroxytryptophan, and they are producing opioid receptors. and they may produce in the presynaptic and also in the postsynaptic part. Postsynaptic part. Different ways. There are different types of receptors. Mu receptors, kappa receptors, and delta receptors. And here mainly the mu receptors are active. And in that case, the opioid receptors, they are also GPCR, G protein coupled receptors, having the serpentine types of, and they have different way of action. They in this case, they reduce the calcium ion entry. At that place, they may have, that is this calcium ion entry can be reduced from this type of substance. And whenever this, then they have their effect. Or in some cases, in this part, that is the postsynaptically, they increase the potassium conductance. So it will hyperpolarize this. And in this case, calcium entry is inhibited. Yeah. And in this way, neurotransmitter is less amount is liberated. And as the potassium ion entry in this part is, and as this potassium, then it is increased potassium transfer, and so it is hyperpolarized. It is difficult to stimulate them. In this way, they have their effect. But textbooks, they do mention that everything in detail is not known, but they have some suggestion, they have their effect. Now, these opioid receptors are present in the higher functions also, and you see a descending fiber, and this, when it is injected into the brain, they do have their effect. Suppose in stress analgesia, then whenever individual is in stress, as in the war field, and then it is possible, that you will feel no pain during the fight, even if injured. And it is the stress analgesia, and so higher fibers are coming on, and they have their effect upon the periaqueductal grade, and from which fibers will descend, and they have their effect upon some raphe nucleus, and over which it affects, and from the raphe nucleus, 5-hydroxytryptamine are liberated. Similarly, the descending fibers, they do have effect on local ceruleus and other neurons of the medulla, from which catecholamines are liberated, norepinephrine liberated, and they somehow have their same effect upon this type of cells in the dorsal root. It is the endogenous opioid system. মোটামুটি আমি তোমাদের একটু ক্লিয়ার করে দিলাম, বাকি তোমরা বই থেকে পড়ে নেবে। আমি খুব টায়ার্ড বোধ করছি, 1 ঘন্টার উপরে 1.5 ঘন্টা হয়ে গেছে, তাহলে আজকে এই অবধি থাক, তোমরা নিজেরা পড়ে নেবে। আর পরীক্ষার জন্য এই পরীক্ষা অনুযায়ী রেফার্ড পেইন্টটা খুব ইম্পর্টেন্ট। ওই ছবিটা দিয়ে কি করে সেটা করছে ডার্মাটোম সেগমেন্ট, এটা ভালো করে তোমাদের পড়ে নিতে হবে। এইটা তোমাদের আলোচনা করলাম, এতটা তোমাদের ডিটেইলে কেউ কোনো পরীক্ষায় জিজ্ঞাসা করবে না, কারণ যারা আছে তারা জানে যে এগুলো এখন ইন ইভলিউশন, কাজে এখন তোমাদের জিজ্ঞাসা করে তারা বিরক্ত করবে না। তোমাদের গ্রস জিনিসটা জিজ্ঞাসা করতে পারবে, কোন ট্র্যাক দিয়ে যাচ্ছে, এবং এটা যে অন্যান্য যে এফেক্টিভ যেটা, এটা কিছু জিজ্ঞাসা করতে পারে। ওপিওড সম্বন্ধে জিজ্ঞাসা করতে পারে, মেট এনকেফালিন বলতে কি বোঝো, ওপিওড বলতে কি বোঝো, তুমি এখানে বলবে মেট এনকেফালিন, লিভ এনকেফালিন, দেন দে উইল বি স্যাটিসফাইড। মোটামুটি আমি তোমাদের বোঝাবার চেষ্টা করতে পেরেছি, এবং তোমরা এটা বাদ বাকি পড়ে নেবে। ঠিক আছে, ধন্যবাদ।